Dr. Smith. This is Mackenzie Martin. I'll be recording my inner life of the cell extra credit and stopping at all the notable points to make sure I get all the information across. Alright, so first, blood flow carries red blood cells down the pathway here, while leukocytes are rolling down the endothelial cells that line the pathway. Leukocytes stick to endothelial cells due to old bonds being broken at the same time bonds are created. The extracellular matrix of both cells are shown sticking to each other. Lipid rafts shown here, which explain the transport of cholesterol from the Golgi apparatus to the plasma membrane. They float across the outermost part of the lipid bilayer. Outside the raft where chemokine has been secreted, the chemokine swings down and presents itself to a leukocyte membrane receptor, which is purple. These seem to trigger cell signaling. Now this is just representative of the inner lipid bilayer. Here you see the red blood cell cytoskeleton, and it's arranged into a network that is made to secure membrane proteins. Actin filaments are shown inside microvilli. These are stabilized by the crossing of proteins. Further into the cytosol, filaments tend to grow in a different direction than the plasma membrane. This is done by adding actin monomers. The actin network is shown with polymers being created and disassembled. Severing proteins wrap around and create kinks in the filament, creating shorter segments that will depolymerize very quickly and create new filaments. The cytoskeleton here is comprised of microtubules and are formed by the polymerization of tubulin dimers. Rapid plus-end depolymerization occurs when the plus ends of microtubules extend towards the plasma membrane. The vesicle is being pulled to and from the plasma membrane along microtubules. Vesicles move in this direction due to motor proteins that link vesicles and microtubules. Membrane-bound organelles are shown here in the cytoskeleton and cytoplasm. The fibrous centrosome is focused on because this is where all microtubules are created. It also contains two centrioles. Nuclear envelope pores shoot up and into the cytosol. They contain both mRNA and proteins. Ribosomes appear to be threaded, translating the mRNA molecules into proteins. Proteins either stay in the cytosol and others are imported into mitochondria, as shown, or other organelles. Several ribosomes dock to protein translocators, which are located on the endoplasmic reticulum. Proteins pass through aqueous pores in the translocator. Proteins then build up in the endoplasmic reticulum's lumen. Other proteins are embedded in the endoplasmic reticulum membrane. The proteins move towards the from the endoplasmic reticulum to the Golgi apparatus with vesicles along microtubules. In the endoplasmic reticulum lumen, protein glycosylation occurs. Now, when proteins are glycosylated, they are transported to the plasma membrane. Here is a vesicle fusing with the plasma membrane, secreting proteins from the vesicle's lumen. That's the first thing that comes out. Other proteins that are embedded in the vesicle's membrane diffuse in the cell membrane. G proteins are activated, which activates integrins, lipid rafts, as pictured. A conformation change occurs in the activated integrins. This change allows them to interact with ICAM proteins, which are nested at the surface of endothelial cells. Here, a leukocyte rolls along an inflammation site. The leukocyte seems to dissolve, which is caused by signaling events that reorganize the cytoskeleton and spread the leukocyte out. The leukocyte will insert itself, one side of it will, in between the endothelial cells. The leukocyte then migrates through the blood vessel into inflamed tissues. Thank you.